now we have set the scene with the physical and chemical properties of the neurons, and we have resting membrane potential. So we have an electrochemical gradient. We have the cell membranes separating charges on the inside and the outside of the cell. And now we're going to find out what happens to cause a neuron to fire. Okay, so the learning objectives for this are what effect do axon diameter and degree of myelination have on the speed of nerve impulse propagation? What are the two types of local potential summation that can result in depolarization of a membrane to threshold level? And how do excitatory postsynaptic potentials and inhibitory postsynaptic potentials interact to determine whether an action potential will be generated or not? So let's get into it. So local potential is the initial change in membrane potential on a neuron, and it's small, and it passively spreads only a short distance, 1 to 2 millimeters. It's sort of the jump start for action potential. So the conduction of information originates with a local potential at the receiving site of a neuron. So sensory neurons are the receiving sites for sensory receptors, and motor neurons and interneurons their receiving sites are on the postsynaptic membrane. So there are two different kinds of local potentials. So a receptor potential is generated at the peripheral receptor of a sensory neuron. So if you bump into something or touch someone, you deform the cell membrane, and you open modality-gated channels on that sensory neuron in order to generate a local potential. That's a receptor potential. So it's the receptor potential is generated at the end receptor of a sensory neuron, and so it's a muscle spindle or a sensory receptor, and that local potential opens modality-gated channels to allow ions to travel across the membrane. Synaptic potentials are generated at a postsynaptic membrane. So those are usually ligand-gated channels in the postsynaptic membranes on motor neurons or interneurons that are stimulated by neurotransmitters binding to them. And we'll look at the whole sequence in a couple minutes. So local potentials can only spread passively, and they're confined to a very small area of the membrane. So there's somehow we have to get enough local potentials together to generate an action potential, so it's going to spread down the entire membrane. So the peripheral receptors, like I said, have modality-gated channels. When local receptor potentials are generated, when the peripheral receptors of a sensory neuron are stretched or compressed or deformed some way or exposed to thermal or chemical agents. Local synaptic potentials are generated in motor neurons and interneurons when they're stimulated by input from other neurons. In other words, neurotransmitters binding to their ligand-gated channel receptor sites. So local receptor potentials cause modality-gated channels to open, and they encode that sensory information into a flow of ionic current. So that's one of the things that neurons do, is they encode information, they transduce it into electrical energy that the nervous system can understand. So in order to get enough local potentials to generate an action potential, we need to add them all up, and there are two different ways. Temporal summation is the combined effect of a series of small potential changes that occur within milliseconds of each other. So temporal summation has several rapid successive inputs that result in a significant depolarization of the membrane. The action potential is a much larger change in membrane potential than the local potential, so we need to get a buildup of these local potentials in order to generate an action potential. The other way we can get a buildup of local potentials is by spatial summation. So the first one is temporal summation. Spatial summation is the summation of several different inputs that results in a more significant depolarization of the membrane. So the strength of a local potential can be increased when synaptic potentials are being generated in different regions of the neuron and they're all added together. Remember how we said some of those spinal motor neurons and Purkinje cells in the cerebellum receive thousands and hundreds of thousands of inputs? So that's spatial summation. Lots of different inputs into the same neuron to get a more significant depolarization. So an action potential, the local potential is a small depolarizing signal that travels passively about 1 to 2 millimeters. The action potential is a large depolarizing signal that's actively propagated by repeated generation of a signal, and the information is transmitted longer distances than just receptor or synaptic potentials. Because the receptor or synaptic potentials spread only short distances, the action potential is essential for rapid movement of information over the long distances. So because action potential is a much larger change in electrical potential, it can be repeatedly generated along the length of the axon. It can spread longer distances than the local potential, and it's actively and passively propagated. So with an action potential, it's an all or nothing. You have to have a sufficient level of depolarization for the generation of an action potential, and that is called the threshold level. So every time sufficient stimuli are provided, the action potential will be produced. Stronger stimuli produce action potentials of the same voltage and duration as the minimally sufficient stimuli. So if you've hit threshold, boom, there goes your action potential. The meaning of the signal, we don't know it yet. It's going to be determined by whatever neural pathway it takes. So the action potential is just you have sufficient stimuli to generate the action potential, and then it propagates down the neuron. The threshold stimulus intensity is the intensity sufficient to produce an action potential. Usually it is a 15 millivolt depolarization, which is a change in the membrane potential from minus 70 millivolts to minus 55 millivolts. That is typically sufficient to trigger an action potential. So bigger than a local potential, but you need at least that amount in order to stimulate an action potential. So this is sort of the path of local potentials and action potentials adding up. So the local potential is the initial change in membrane potential, which spreads only a short distance along the membrane. So you get the deformation of a receptor membrane, which generates a receptor potential. If you get enough of those, you can generate an action potential, which results in depolarization of adjacent membrane areas, and it propagates down the neuron. So the action potential is a brief, large depolarization in electrical potential that's repeatedly regenerated. So it propagates down the neuron, it depolarizes the presynaptic membrane, it releases neurotransmitter, the neurotransmitter diffuses across the synaptic cleft, it stimulates receptors on the postsynaptic membrane, ion channels open, it generates a synaptic potential. If there are enough of those synaptic potentials, you can generate another action potential which propagates down the next neuron, which causes depolarization of presynaptic membranes, release of neurotransmitter, and it goes to the next neuron. So each time you reach a synaptic cleft, it's a chance either to amplify the signal or diminish the signal. Okay, keep that in mind. So the action potential is produced by a sequence of 3 events: rapid depolarization, and the voltage-gated sodium channels open, because the change in voltage opens the sodium channels. Then the channels close, and it causes a decrease in sodium ion conduction. And then rapid repolarization, which is when the voltage-gated potassium channels open. Because of the voltage, or because of the ion chemical gradient, when the channels open, the ions will move in the direction of their chemical gradient. So there's naturally more sodium outside the cell and more potassium inside the cell. So when the sodium channels open, sodium rushes into the cell, and then they close. When the voltage-gated potassium channels open, potassium goes out of the cell, and then it closes. So the change in membrane potential happens with the movement of those ions. The refractory period is a period of hyperpolarization during which the membrane potential is even more negative than the resting membrane potential, and it's difficult to initiate a subsequent action potential. It's really, the refractory period is really defined by the ion channels. Some of the ion channels become inactivated immediately after opening for an action potential, and they require a specific amount of time before they can be activated again for a subsequent action potential. So I say it's like the gated community where one person uses their key card, the gate opens, they drive their car through, the next person has to wait until the gate is ready before they can use their key card to then open it. So only one car can get through at a time. That's the analogy. So the active component of an action potential, the local potential creates the threshold stimulus, or the summation of local potentials. Voltage-gated channels open and generate an action potential causing a rapid influx of sodium. That's the depolarization of the cell. The inside of the cell becomes less negative because those positive sodium ions are going in. The sodium channels close, causing decreased movement of sodium. The voltage-gated potassium channels open, and that's the start of repolarization. Potassium flows out of the cell, causing hyperpolarization, which is where it becomes even more negative than resting potential. And that is the refractory period when it's harder to depolarize the cell. So there are two distinct refractory periods. The absolute refractory period is when the membrane is unresponsive to stimuli. So this is because the sodium channels are still regenerating. They're still reorganizing so they're ready to be opened again. So during the absolute refractory period, you just cannot open those sodium channels. During the relative refractory period, the membrane potential is returning to the resting level, and it may be hyperpolarized, but you could generate, if you had a big enough change in potential, you could generate another action potential during the relative refractory period. So if it's a stronger than normal stimulus. So during the absolute refractory period, those membrane channels are just getting ready to work again, but they're not ready. During the relative refractory period, they're ready and it's getting there, and if you have a bigger stimulus, you could generate an action potential. So in terms of propagation of action potentials, there's a passive component and an active component. So some axons just may be specialized for faster action potential progression because they either have an increased diameter of the axon or increased myelination of the axon. So you can think of it with the increased diameter, a wider hose will pour more water in less time. And you can think of the myelination as like tape around a leaky hose that concentrates the flow. So the change in electrical potential in the passive component of propagation The electrical potential spreads passively along the axon to the adjacent region of the membrane. And when it reaches the threshold, another action potential is generated and continues spreading along the axon. So the depolarizing current passively spreads down the axon, causing the interior of the axon to become more positive or less negative, if you want to think of it that way. In the adjacent membrane, when the depolarizing current reaches threshold level, sodium channels open, causing rapid depolarization of the membrane. So it's those voltage-gated channels. The action potential is generated and the depolarizing current continues to propagate down the axon. So to keep the amplitude of the action potential above threshold, the membrane potential cannot be allowed to return to the resting potential. So you need to continue to have that stimulus. Myelin increases the speed of the action potential and the distance the current can passively spread. The myelination is that sheath of protein and fat surrounding the axon. It provides insulation and prevents current flow across the membrane. It increases the speed of action potential propagation and the distance that a current can passively spread. Thicker myelin leads to faster conduction and greater chances for action potential propagation. So there are spaces between the little bundles of myelin that are called nodes of Ranvier. They're small patches of myelinated axons which lack myelin, and they're specialized for active propagation of action potential by allowing the ion flow across the membrane. So they're every 1 to 2 millimeters along the axon, and they contain high densities of sodium and potassium channels, those voltage-gated channels. So when the action potential spreads rapidly along the myelinated region, it slows when it's crossing the nodes of Ranvier. And they call it saltatory conduction, where the action potential quickly jumps from node to node down the myelinated axon. So it sort of covers more space in the same amount of time. There are those 3 functional groups that we talked about of neurons based on direction of flow. Afferent neurons carry sensory information from the peripheral outer body towards the central nervous system. Efferent neurons relay commands from the central nervous system to smooth and striated muscles and to glands. And interneurons are throughout the nervous system, processing information locally or conveying information short distances. Interneurons are the largest class of neurons. The interaction between neurons, you can have divergence and convergence, and they contribute to the distribution of information throughout the nervous system. With convergence, multiple inputs from a variety of cells terminate on a single neuron. With divergence, a single neuron with many branches can terminate on a large number of neurons.